field of theoretical and computational neuroscience with a background in biomedical engineering, and her research focus on understanding the brain dynamics, the functional connectivity, but also structural connectivity. She did really in our community a very nice work to advance the analytical tools and large-scale computational brain models to investigate the mechanism underlying the function, but also underlying the structures. So this is a very important input. She contributed in the field with, for instance, LiDAR algorithm, which identify features to understand the whole brain dynamic related to cognitive and behavior conditions. And she is recognized as an important person in this field and in the field of neuroimaging. So her research is really bridge between mathematics, computational neuroscience, and also application, and mostly to understand the brain function in normal subject, but also in case of diseases like psychiatric disorders. She received a prestigious award from L'Oréal, Award of Women in Science in Portugal. And her research provide really a new vision, a new understanding of the brain function at the macroscopic level. So, Joana, it's a pleasure having you with us today, and thank you very much for accepting our invitation. Thanks.
So, hello, everyone. Thank you. Thank you for the really kind introduction. It's always a pleasure. Yeah, I really regret not being there in presence, but I hope I appreciate the opportunity to present remotely, and I hope I will be clear and my message will be transmitted. So in this first session this morning, I will present about synchronization mechanisms in the brain space-time connectome. It's more focused on EEG activity, and in the second session this afternoon, I will focus more in fMRI. So overall, I'd like to start with a general motivation slide. So we want to understand, right, brain function, and our best correlate with brain function appears to be nearly infinity of activity patterns we detect across modalities from the brain that relate to cognition, emotion, and behavior. But now when this function is impaired, we have cognitive impairment, psychiatric disorders, disorders of consciousness, and this is a real problem, right? We need to understand what is going on so that we can try to develop better strategies to fight these disorders. So how do we do this? What are the fundamental principles? What are the mechanisms, the constitutive equations? So these are still unknown fields. And the best we have is to look at the brain structure, right? To understand how we can go from structure to function. And when we understand this, we may find better solutions to fight these disorders. So when we look at the brain, we know it's composed by 100 billions of neurons, not only neurons, but also glia cells, fluids, ions, neurotransmitters. And these are connected by 100 trillion synapses, right? So it's perhaps one of the most complex networks in nature. So it's very interesting to approach it from a network perspective. So nowadays we have access to the so-called structural connectome that can be obtained in vivo, non-invasively with diffusion MRI. And this is composed by a network of white matter fibers that connect distant brain areas. And the question here, so we understand that these connect brain areas, so the information can, or the signals can pass through these wires, but how far does this connectome structure shape brain dynamics? So to understand how a network structure can shape brain dynamics, this is a networked dynamical system. So I made a small cartoon just to see. So if we have one dynamical system that is described by its own set of differential equations, when it's perturbed, it will respond. But now if this is connected to other dynamical systems, right? When we perturb one, this activity will propagate to the rest of the system in a way that the behavior of each unit can only be understood if we understand the whole network dynamics, right? So this is a networked system in a way. And this network structure shapes the dynamics, means that the activity in a single node of the network cannot be understood without considering the dynamics of the whole network system. So there is a lot of studies, here I name a few, showing that network dynamics actually affects the network structure actually affects the dynamics. There are synchronization phenomena that are studied. People have reported that there can be metastable chimera states, meaning that some subsets of the system may synchronize while the others are desynchronized. Different dynamics can emerge from different properties of the networks, if the networks are small world, if they have specific clustering or modularity. But above all, there is this concept of critical phenomena that are observed in complex networks. And this is an active research in the fields of mathematics and physics, right? So when we have this network of lots of neurons and synapses, and we want to understand how this gives rise to these activity patterns, we need a dynamic model, right? To investigate how the network structure can then impact the macro scale features we observe in brain activity. So in terms of what is a dynamic model, so a static model can be a model just of the anatomy, right? But then a dynamic model considers time, so how things evolve over time, and this means that the values or parameters are time dependent, and for that we need differential equations. This is a model here, this illustration is just from a single neuron. And at the level of dynamic models, we need to understand how to go from the micro scale of a single neuron to the macro scale of the whole brain, right? Because this is it's at this level that we are finding strong alterations in neurological and psychiatric diseases. So on one side, we have the signals that we detect with EEG and MEG that reveal collective rhythms. So rhythms that are generated by millions of neurons together and that reveal macroscopic patterns that can be detected at the scalp level. And with fMRI, which I will focus on in the next session, we observe these long-range correlations in activity patterns. So how to address these problems? So there are different dynamic models that can be used across different scales. So this is just a cartoon, but to explain that we can either go from the microscopic level of models of synapses at the nanometer scale, or we can have really the cellular level description of the system. Then we can have mesoscopic models that consider a single, like a population of neurons. This can be a population of homogeneous neurons, or we can have models of interacting neuronal populations, or even include multiple layers. And then at the macro scale, we can consider what is the behavior of multiple cortical columns and use field models, and ultimately go to the whole brain level where we consider larger brain areas. So this is again another figure to show how when we consider how the different cortical columns or even larger brain areas are connected through these long-range white matter tracts, it's important to consider that each parcel is densely packed and composed already by millions of neurons and billions of synapses. So there are three major ingredients that play a role in shaping the brain network dynamics, right? We have on one side the structural connectome, which we can have at the voxel level resolution from tractography, like the video we saw before. But then we can reduce this because it's very computationally expensive, and at this stage it's still hard to run simulations at very high resolution. But we can parcel the brain, let's say divide it in, for example, 100 brain areas, and then count the number of fibers that detect it, connect each pair of brain areas. So here, for example, we see an example of a brain network model where each node represents one anatomically distinct brain area, and then the link, the connection between these areas is scaled by the number of fibers that are detected between them, and also by the distance, right? It's also important to know. So we can represent this in the matrix format, where for each brain area, we see how strongly it is connected to all the other brain areas P. And then we can also have a distance matrix, which is, for example, taking into account the length of the fibers between brain areas, because as we will explain, the distance can also have a strong impact. So then the second ingredient in these models is the node dynamics, right? So in each node, which, as we said, is a dynamical system already composed by millions of interacting neurons, and we can have different levels of description for this node dynamics. We can either use those detailed cellular models that consider really the spiking activity of the neurons, or use more reduced models of neuronal populations, like in this example is the Wilson-Cowan model, already developed in the 70s, to explain how excitatory and inhibitory populations of neurons can interact together to generate oscillatory rhythms. And then we can have even more abstract models that can just represent the oscillatory dynamics of a given brain area, considering like just oscillations in the local field potential. I will go on in detail about this. And then we need to consider also the coupling function. So it is how the activity of a node is affected by the activity of the other nodes, like we said. And it's important to consider also that time delays exist, right? So the propagation will not be instantaneous, the transmission, and the delays are not homogeneous. So this can also strongly impact the dynamics and enrich what we will be seeing. And then, of course, we'll have external properties such as noise or physiological rhythms that can act on this network. But overall, this is the general picture. So each brain area represents a node in the network. And then depending on how strong is the coupling, this will scale a coupling function, which is how connected brain areas interact together. So at the node level, we consider we detect oscillations in the local field potentials. And neuronal populations have the ability to engage in oscillations in the gamma frequency band, where 40 Hertz is usually the typical frequency that is recorded. And it comes from the balanced interactions between excitatory and inhibitory neurons. This was proposed already in the 70s, this model, and in the late 90s, it was demonstrated that even if you just take a piece of cortex and in vitro, they are able to exhibit. So if you just put an electrode inside the tube and with respect to a reference, we observe that there are oscillations in the local field potential that are significantly self-sustained. They are not fully self-sustained, but they can be approximated to an oscillator. And the question is, how do these local gamma band oscillations organize when they are embedded in the brain network, right? This is the main question that arises, and how are they related with the slower rhythms that we detect with EEG? Because in EEG and MEG, it's harder to detect this, so the rhythms that are associated with disease are usually at slower frequencies. So how we reduce, this is just to explain or to illustrate how we reduce like the local field potential oscillations at 40 Hertz to a phase oscillator. So basically this is how it can be represented in the unit circle as an oscillation at 40 Hertz. So this is an abstraction, of course, it's a huge reduction, but it helps to then try to understand more the fundamental principles that are behind these things. So now I prepared here also an illustration, an animation to explain what happens when you have oscillators interacting together. So if you have two identical oscillators, like two 40 Hertz oscillators, but they are not connected together, they are not seeing each other or feeling each other, each one will evolve forever at its own natural frequency. But now when they are coupled together, it means that they interact with each other. They can synchronize together. And this synchronization is predicted by the Kuramoto model of coupled oscillators. And basically, it says that each oscillator is sensitive to the phase difference with other oscillators. So each node N has its own natural frequency, and then there's an interaction term that is scaled by a given coupling strength K, and then it depends on the difference between the phases of the two. So it always tries to minimize this phase. But what happens if there is a conduction delay? As you saw now, the circle started going slower, right? So if there is a time delay in the equation, which means that each node only feels the other after a certain time point, the time delay slows down the frequency of synchronization. And this has been demonstrated analytically. So in the work by Niebuhr and colleagues in 1991, and what they showed is that the frequency of this collective oscillation, so like I put here several oscillators coupled together, and then the large circle represents the collective, the system, the whole system oscillation. And this collective frequency depends on the mean natural frequency of all the oscillators, on the mean time delay between them, and also on the size of the system. So as we see from the equation, the larger the N, or the larger the K, or the larger the delay, the T, they're always in the lower part of the equation, of the fraction. So this will always reduce the collective frequency. So this is relevant for systems with large N, large number, right? So usually people do a simplification where the synchronization frequency would be the same as the natural frequency of the oscillators. But if we have time delays that even if they are very small, but we have a very large system together, this phenomena happens. Also then, another particularity is that if the system, if the network has a complex topology, which means that they have like clusters or modules, it can happen that for a certain range of parameters, if the coupling is sufficiently strong, only a subset of the network will synchronize together. So this can create like small cluster frequencies, right? And this partial synchrony may be weakly stable, meaning that like a subsystem may synchronize and then desynchronize, and then synchronize and desynchronize. So this can lead to fluctuations in the power of these cluster frequencies, right? So now this mechanism that exists, right? It's a fact and it's solved analytically, but we don't know if this can be applied or explain what we observe in brain activity. So the idea to test is, could this mechanism explain the transient emergence of oscillations that we detect at the macro scale in MEG and EEG sequence? So for that, we need to test this hypothesis. We need to run simulations and compare with experiments. So we can have the brain network model. We place at each node an oscillator, so it resonates at 40 Hertz, let's say, and then we couple them together with a distance proportional to the diffusion track. And they are also, it's also important to since there is no directionality of the fibers, we don't know, we consider that this coupling is reciprocal. And so how to incorporate the connectivity in a brain network model? So using this Kuramoto model of coupled oscillators, which is a simple paradigm to study synchronization phenomena, we have the normal equation of Kuramoto, but what we did is to introduce in the interaction term, so we have a global coupling strength that scales how much each unit receives from the rest of the brain, right, of the system. And then here we weight the coupling by the connectivity between each brain area. So the weight, the number of fibers, for example, that are detected between areas. And we also introduce a conduction delay, where the conduction delay is proportional to the distance. So we assume homogeneous velocity, because in reality, we still don't know exactly what is the time between interaction between areas, but we do this approximation. And the advantage of the Kuramoto model, as probably most of you know, is that there are many analytic solutions, meaning that some phenomena that are observed can be solved mathematically. And also we can easily run computer simulations and study the role of the connectivity. So, yeah. So what we did was to take, for example, the MEG signals that are recorded. This was data from the Oxford Center for Human Brain Activity from 20 subjects. So we have the signals in the sensors. They are bandpass filtered in a given frequency band, and then they were source projected into the locations of the different nodes or the centers of gravity, let's say, of each brain area. And so we detect for each brain area, we have a signal in a given frequency band. And then what had been previously observed is that the envelopes, that is the amplitude of these signals, correlates together between brain areas, driving long-range functional connectivity. So distant brain areas have correlated power fluctuations. And this is what is called frequency-specific functional connectivity. So we can have for each frequency band, here we took 10 frequency bands, and for each frequency band, we correlate these envelopes. So we have a matrix of functional connectivity for each frequency band. And then we had the model of coupled oscillators. I will go in detail also about this, but this is like just a graphical abstract where we use, as I said, the Kuramoto model of coupled oscillators, incorporating the coupling weights and the time delays from the space-time connectome, this is what we call, and we ran simulations on it. And then we also bandpass filtered in the same frequency bands. We compute the correlation between these envelopes, and we obtain the simulated functional connectivity profile, right? And then the idea is to try to find if there is a range of parameters where we have an optimal fit between the functional connectivity profile that is observed empirically and the one observed in the simulation in the model. So when we did this, so here I'm trying to illustrate the fitting. So the plot in panel A means as we vary the coupling strength of the system, of the model, right? And as we vary the mean delay, there is a range of parameters where we find the best fit between the real and the simulated functional connectivity. But maybe better to understand what is going on, it's important to look at different properties of the simulated dynamics. So when we have here in panel B is the synchrony degree, or the mean of the Kuramoto order parameter, which means how strongly correlated brain areas are. So for strong coupling strength, like here above 10, the system tends to synchronize together. So you see everything in red. If the coupling is 0, the synchronization is null, nothing is synchronized, of course, because they are initialized with random phases. But then there is a border when we have time delays. So without time delays, the synchrony border is very fast. Like you're either asynchronous if they are uncoupled, as long as the coupling is above 0, the system will synchronize. Now, when there are time delays, it takes more coupling to synchronize, and we have a critical boundary there between incoherence and synchrony, where the synchrony degree fluctuates over time. So this is what we see in panel C. It means that there are strong fluctuations over time. So even without noise, so these are simulations without noise, just 90 coupled 40 Hertz oscillators, and we see that the synchrony degree fluctuates over time. And this generates fluctuations in the envelopes of the oscillations that are detected, like in the relevant range here is between 8 and 30 hertz, so which means in the alpha and beta bands. So it's curious because we only had 40 hertz oscillators, and we see these amplitude fluctuations at slower frequencies that are related to the mechanism I explained earlier. So for example, here is for 10 different frequency bands analyzed, how the functional connectivity matrices look like, and then at the bottom is how the functional connectivity matrices look like from the model. Of course, it's not a one-to-one match, but what is important to see is that indeed not only we see similar patterns that come, of course, from the structural network. So we see when it's more red, it means that the brain areas are more correlated together, and we see stronger correlations in this alpha and beta bands, let's say. So the values are stronger. So what is happening? So if we just look at the signal, as I said, like an oscillator just has a phase. So here we just plot the sine of the phase to see it over time, or like the real part or imaginary part of this signal. And if we look at the simulated signal and the amplitude doesn't fluctuate, right, in a Kuramoto model. So it's always between -1 and 1, it's the sine of a phase. And if we correlate these signals for a certain time, the correlation here is very, very low. But if we look carefully, if we then filter exactly the signal that I have on the top, but filter it in a given frequency band, we see that from time to time, the oscillators slow down, right? So we see that there's a point like after 0.5 seconds where we see a slowdown in the periodicity of the signals, and this is captured when we filter the signals. So this induces an amplitude modulation, right? And this amplitude modulation now correlates like almost 80% between these two brain areas. And if we look at the 40 hertz, at the frequency of the natural frequency of the oscillators, if we filter around that range, the correlation is again almost null. So there is a frequency-specific correlation that can be explained by this model. And this correlates with the degree of synchrony over time. So now what I'm plotting here is over time, over like 300 seconds, the bandpass simulated signal in blue. And then we have the envelope that is slowly low pass filtered. And we see that from time to time, we see there is an increase in the envelopes. So the power increases and decreases. So if we look at the spectrogram, let's say, so if we look at the frequencies on the y-axis over time, we see that most of the power is around 40 hertz, which is the natural frequency of the oscillators. But then from time to time, we clearly see there's an increased power in these low frequencies. And these low frequencies are strongly, the appearance of these frequencies is correlated with fluctuations in the synchrony degree. What does this mean? It means that when the synchrony degree increases, right, it means that those oscillators will synchronize together at a reduced collective frequency. So we see the collective frequency appear, and this drives the amplitude envelopes in the signals that we simulate. And this is one important thing, is that this is simulated without noise, right? That's why we call it metastable, because the synchrony is not, or the switch between synchrony and incoherence is not driven by noise, but by the complexity of the system, let's say the heterogeneity of the system. But it also survives with noise, so we can add noise on it, but it's not noise that is driving the transitions between synchronized and desynchronized states. But then this was a model, again, it's just with limit cycle oscillators, and there were like, in general, there was a general comment asking, yes, but it works with, that's for limit cycled oscillators, and in the brain, in reality, the local field potentials are not always oscillating at 40 hertz. The oscillations are damped. So what this means is that the gamma frequency oscillations are not in a limit cycle, so they can be represented by a damped oscillator in the sense that it drives an oscillation and then the amplitude decays back to zero over time. So it's like an oscillator in the subcritical regime. And for that, we need a different type of equation, which is not anymore the Kuramoto model, but we can use the Stewart-Landau or the Hopf bifurcation model. That is an equation that, depending on the parameters, can exhibit different dynamical regimes of the oscillation, right? If the gray dashed line is when the system has no damping, so it means that it will always be in this stable limit cycle. Then if we increase the damping, so if we go to the subcritical regime, the system will just oscillate. When it's perturbed, it will oscillate for some time and then decay in amplitude. This is the green pattern. And then if it's over damped, we perturb it and it just decays down to equilibrium. So this equation allows to have these three different regimes, or if you like to explore them as a continuous. But the interesting thing is that if you have a damped oscillator and just perturb it with a white noise, you see the fluctuations in the amplitude, like I'm illustrating here at the bottom. So we see that the oscillations appear and disappear, and this looks more like what we observe in local field potential recordings. So this is the general form of this equation. So to describe all these systems together, we have this Stewart-Landau equation that has a parameter A here. And depending on how the value of A, so if A is positive, we are in a limit cycle, which approximates the Kuramoto phase oscillator. And then if A is negative, it means that the system is damped. And there is a range, like here, the red range, which is the subcritical range, where when it's perturbed, it still exhibits an oscillation. Then if it's overdamped, if the A is too negative, then the system will not even display any oscillation. So this for the single node. So the idea also of, so Gustavo Deco suggested this solution to explore this was to use the Stewart-Landau equation and to couple it with time delays and see if the same phenomena we were observing in the Kuramoto model can still survive, right? Because this had not yet been demonstrated. So we set the A parameter, so the distance to the bifurcation, let's say, to -5, such that the 40 Hertz oscillation decay after around 500 milliseconds, because this is the time scale of the inhibitory time scales of the concentration of inhibitory neurotransmitters. And then we have these coupling terms. So instead of just coupling the sign of the phases, minimizing, we just do a difference coupling, but these values are in the complex domain. So it's a difference between two complex values that have both a phase and an amplitude. And they are coupled with time delays, of course, like we wanted. And of course, we need to add white noise. So we add uncorrelated white noise, and we perturb all units with a bit of noise. Because if we don't, the network is simply, since all units are in a subcritical regime, if there's no perturbation, it's just off. It's just in a static point, right? So when we couple these units together and we perturb them with noise, what we find is if the coupling is very weak, the oscillation, the signals that we simulate, we observe like before. So this is just a damped oscillation perturbed with noise. It will oscillate close to its natural frequency. There are still some coupling effects that induce a shift in the peak frequency, but the synchrony degree is low, is always close to 0. It oscillates between 0 and 0.3. So the system is weakly coupled, but not enough coupled to induce these collective oscillations. Now, when we have strong coupling, on the other hand, on the right side, we can have the simulated signal. What we observe is that the synchrony degree can go up to 1. And when it goes, like when it synchronizes, we see strong low frequency oscillations. So the system, instead of being with a peak at 40 Hertz, we find that it goes down to 2.5 hertz. So this aligns with what we were seeing already with the Kuramoto. And if we have an intermediate coupling, because this is the range where, the critical range where the brain is believed to be operating between incoherence and synchrony, right? In this intermediate range, we observe that there are strong fluctuations in the synchrony degree. So you can see in the plot, it goes from almost 0 to almost 1 and almost 0 to almost 1, never reaching 1. And when there is an increase in the degree of synchrony, we observe oscillations. In this case, these ones are, for example, at 7.5 hertz. So it shows that with the same system, again, 40 hertz oscillators, this time they are damped, but they are perturbed with noise, we observe this frequency modulation even at the single node level. Now we take, but could still this equation that was predicted for the Kuramoto model for collective frequencies predict the frequencies that we are detecting in the simulations with the Stewart-Landau equation? So if we just solve this equation, we just calculate what would be the collective frequency for the different ranges of parameters. So we have values for all the parameters, and we can compute what would be the predicted global frequency of the system. And here I put a line to show where the 10 hertz, because 10 hertz is the oscillation that is mostly detected from the brains at rest, from human brains. And we find that indeed, for a certain range of couplings and a certain range of conduction delays, we predict that our system will be able to synchronize at this frequency. So what we did is for each value of coupling strength and conduction delay, we compute the power spectrum of the simulated dynamics, so the mean for all the nodes, let's say. And then we show here on the right side, we have two plots of the peak frequency. In the left one is the simulated, the peak frequency of the simulated data. And on the right is the predicted global frequency. And the predicted global frequency doesn't have any sensitivity to the coupling. It assumes that the system is synchronized. But in the real data, we find that there is a border, right, below which everything is at 40 hertz. And this is the border of synchrony. Let me see. Okay, so if we look at the synchrony plot below, so above this line, below this line, the system is not synchronized. So everything will still be at 40 hertz. So that's why we see here all red in the global peak frequency. But above, once the system is just a little bit synchronized, we immediately see a peak at the level at the predicted collective frequency. So this is a concept that may not be very trivial, but it's very interesting to see that the simulations with the Stewart-Landau oscillators still can be explained, or the results can be explained with solutions that were derived for the Kuramoto model alone. And so then we try to find where in the parameter space we had the best fit to average MEG power spectra that we detect. So we had power spectra from 80 subjects, so MEG data from 80 subjects from the Human Connectome project, an online database. We just downloaded the power spectra, let's say, and we averaged across all the sensors. So here we plot all the power spectra, so it's just normalized. It's not in logarithmic scale, but really it shows that most of the subjects have a peak here around 10 hertz, and this is the average one, the green, the blue line, let's say. And we see where our model best approximated this power spectrum. And then we did this also for, so the colors here showed it's a difference measures. So the blue means that we have a better fit, right? And in red, we have a bad fit. So if we don't have delays, we never have a good fit. If the coupling is not sufficiently strong, also we never have a good fit because the system is not synchronizing. But we had the best fit with all the 89 participants in an area of the parameter space, right? So for sufficiently small conduction delays, like from 2 to 10 milliseconds, mean conduction delay. So this is the mean of all. Of course, they are heterogeneous, but the average conduction delay, and for a sufficiently strong coupling strength. And this is where also where we had the best match is where the global frequency approximated the 10 hertz, right? So it's here. And now I want to show, so here is, for example, the case where we didn't have any delays. So the delays are 0. It's important to consider that many models, to simplify, they simply don't consider time delays. But time delays are an ubiquitous property in nature, right? We don't have anything cannot be exactly in the same point in space. So there's always a distance between any type of unit, and this distance will always be associated with a certain time. In addition, even when we think we are not considering time delays in the models, if we are integrating them over time, the time delay is always the time step, right? In the next time step, this unit will respond to the past of this unit. So there's always the dt is always a time delay. And this is very important in the simulations because we need to simulate with very, very small integration times. We went to 10 to minus 5 seconds, right? To simulate this properly because the time delays are very small. But anyway, without time delays, we don't have any collective oscillations. All we have is 40 hertz oscillations with noise. But then if we add delays and have sufficient coupling, we observe that there are these collective oscillations that appear in the simulation. So let me just also explain this plot. On the left side, so on the y-axis, we have each brain area, let's say, or each node in the network. There are 90 nodes. And then we show the simulated time series, filtered below 30 hertz because we wanted to exclude the 40 hertz, which is still there, but will be out of phase. And below 30 hertz is what we detect mainly in EEG and that. And then we add a shade, for example, in blue. What we see in blue, it means that there is an oscillation there is power, let's say, between 8 and 13 hertz. 5 standard deviations above the baseline I showed before. So the baseline is when we don't have delays. We define what is the standard deviation of that. And then here we show that adding couplings and with time delays, these oscillations. Another very nice feature from this plot is that whenever, most of the time, when we see an increase in the power in the alpha band, so blue, let's say a shade, it is we see a vertical line. This means that these different nodes in the system exhibit simultaneously an increase in the power at that frequency. So again, it links to synchronization phenomena because they all increase their power in that frequency band. And below I show the level of synchrony, so in red, and also the amplitude envelope of the system. So we see that they strongly correlate together. So more synchrony gives stronger amplitude at slow frequencies. Now, when we look at this in more detail, let's say, how can we characterize these oscillations, these oscillatory modes, these metastable oscillatory modes, we call them MUMs. So one way to try to characterize them was to look at the size, meaning how many units increase their power in this collective frequency at the same time, right? So imagine there will be different nodes that's spontaneously synchronized together at a given frequency. So that is the size of this coalition or of these modes. Then the duration, so how many consecutive time points these oscillations remain active. And we do this for different frequency bands, for the different frequency bands. So for example, for this set of parameters, we can calculate the duration of these metastable oscillatory modes. So we see that they last around 200 milliseconds. They involve more areas. So in terms of size in the alpha band, so we see they involve around 20 areas, but sometimes 40 areas. So there's a large standard deviation. And in terms of occupancy, is also how much of the signal can be explained by these oscillations. So what is the proportion of alpha in the whole simulation or the band limited power. So what is happening here over time? I'll again try to make a cartoon to convey this information. So we have here the level of synchrony over time, and we see that it's fluctuating. So it means that when the synchrony is low, all the oscillators are a bit out of phase, imagine at 40 hertz. And then when the synchrony degree increases, the oscillators become synchronized at a slower frequency, and then it decreases again, and then increases again, and then decreases again. Sorry, I don't know why here is missing, but synchrony. But the thing is that this can relate also to these theories that in the brain we have this balance between segregation and integration, right? So there are periods where the system is more segregated, and then points in time where it's more synchronized together. Now, another nice thing that we find is if we strongly increase the coupling strength between units, right, we force the synchrony. And when we force the synchrony happens at even slower frequencies and involve more brain areas. So in this case, again, the power strongly correlates with the level of synchrony. And when we see here this yellow and purple, so yellow is in the delta frequency range. It's the slowest waves that we detect from our brains, from human brains, and they are particularly detected in slow wave sleep, in deep anesthesia, in coma, in disorders of consciousness. So there are these slow waves that are very macroscopic. So they really have high power in all the electrodes and all the sensors. So this model could also explain that maybe what we're capturing is that the system is failing to segregate. Right, it's more synchronized than it should in some disorders, but of course during sleep that would be natural. Then we also looked at how this drives frequency-specific envelope functional connectivity. Right, in the first column we show if the coupling is weak, we don't have correlated envelopes in any frequency band. Now in the optimal range, we have a pattern of functional connectivity in the beta band, for example, another pattern of connectivity in the alpha range. We see there are more strong correlations in this range, but also in the others, we still detect some patterns of connectivity. Now, if the coupling is too strong, we only have power at very slow frequencies, and those envelopes are also strongly correlated. Right, and then with long delays, the system becomes more asynchronous, and then probably measures of instantaneous, let's say, correlation are not so sensitive because there are more time dependencies between units. So overall, in this from this approach, what we can demonstrate, so we are not demonstrating that this is what is actually happening in the brain, we still don't know, but what we show is even if we have a fixed network structure, so the brain, the connectome, and we perturb it with white noise, then depending on global parameters that can be associated with physical properties, like the global coupling, like could there be some neurotransmitters or chemicals that enhance the coupling strength between units or allow them to behave more independently, or the conduction time also can be associated with properties of the availability of ions, or even with the axonal, the myelinization of the axons, let's say. But what we find is that depending on these properties, we can find an optimal range where indeed we observe these oscillatory modes in the alpha band. And this has been described, the obsession here with the alpha band is that indeed it is a very ubiquitous property from our brains, like it has been described already 100 years ago, that indeed the electrodes that you put here have 10 oscillations per second. And this proposes in a way that there could be just it could be explained by these very general universal principles. So here again, it's the same story, but just to show the plots. So the same network structure, we can have different dynamical regimes depending on global parameters. And of course, then this is very abstract again. I always say this is not a model of the brain. This is a model of a mechanistic principle. But then the idea is that this can then be extended to more detailed models of coupled systems with time delays, right? If this phenomena exists, then maybe if we use more detailed models such as the Fitzhugh-Nagumo or the Wilson-Cowan or other types of models, if we introduce time delays, will we observe this phenomena? And also it can help because sometimes when you have very detailed models, then you run the simulations and you see a lot of things that cannot be easily explained. So this way, if someone sees collective oscillations appearing, they can be linked to this property. And this is something I did also in collaboration with the group of Francesca Castaldo and Francisco dos Santos. And what they did was to first apply this measure of the MUMs, or the metastable oscillatory modes, both in empirical fMRI and in MEG across different frequency bands. And then they tested either the Stewart-Landau equation, like I just showed, or using the Wilson-Cowan model. The Wilson-Cowan model is this model of coupled excitatory and inhibitory populations, and it was tuned to exhibit damped 40 Hertz oscillations, of course, and we observed that with the Wilson-Cowan model, the fit with empirical data was even better, but at least we could explain why this was happening, right? Because we were putting time delays between 40 Hertz oscillators, and this is what was giving rise to these metastable oscillatory modes that are observed in simulations. So overall, in terms of discussion, or the take-home message, let's say, is that these transient collective rhythms, we propose that they are like, we don't propose, this is just a fact not related to the brain, are an emergent property of complex delay coupled systems at the critical border to synchronization, right? So we have a system, any system, if you have a modularity structure and you are in this coupling strength that is not sufficiently strong to induce full synchrony, but only this transient synchronization, you'll see this happens, the appearance of slower oscillations that are collective. And then, yes, we propose that maybe potentially the brain rhythms that are detected in EEG may be a signature of this fundamental property. And also this property may explain the origin of collective oscillations in more detailed computational models. Because of course, I agree that not all brain areas are identical. The assumption of homogeneous velocity speed is also just an assumption. The way the network has very low resolution. So the idea is, if we build up on a more complex model, at least we already know what to be looking for. And yeah, well, thank you for your attention. As I said, I am hosted at the Life and Health Science Research Institute in the University of Minho in Portugal. I'm more in a group related with psychiatry, from brain to mental health. I also collaborate a lot with the Champalimau Foundation in Lisbon, which is the results I will explain in the next session. I'm funded by La Caixa Foundation. And yeah, I want to thank, these are the main collaborators for this work I was showing here. And thank you for your attention. I have no idea how long. Yeah.
So thank you very much, Joana, for this very nice presentation. We are going to move to the audience for some question, maybe some explanation also. Is there any question from the audience? Yes.
Hello, can you hear me?
Yes.
Okay, thanks a lot for the great talk. It was very interesting. Especially I'm discovering the concept of 40 Hertz oscillator, so it was very new for me. I was wondering, so this 40 Hertz oscillator, is it like a property of the brain? Like, is it very constant, or can it fluctuate between, I don't know, individual or brain region, or is it really something fixed? And
Yeah, that is a good
yes.
question. Yeah. Okay, so regarding that, indeed, it's very curious because it seems that they at least there are papers shown. First, they showed with hippocampus. So there is a paper from Kizan 90-something where they showed sustained 40 hertz oscillators from a piece of, let's say, hippocampus in vitro. Then they tested again with cat somatosensory cortex. Then they tested again with other types of pieces of cortex, right? And of course, it's not, maybe it could be 39, it can be, it's gamma band oscillations, let's say. But so far, at least from the reviews that exist from Lienz and all of that on the generation of brain rhythms, at the neuronal level, we can only explain the gamma band oscillation. So it has never been induced in vitro like 10 hertz, you know, just from a small piece of cortex. It seems that 10 hertz are a network frequency, let's say, or you need, maybe if you had a larger piece of cortex, maybe it could synchronize it at that frequency. But yeah, it is, there is a paper from Brunel and Wang 2003, I think, about what determines the frequency of local field potential oscillations, because if they are driven internally, it means that it's an intrinsic rhythmogenesis, right? There is no external drive for those 40 hertz. It needs to come from there. And that, and also they show that if you change the concentration of the fluid outside, so of the extracellular field, so they need certain concentrations. So those 40 hertz oscillations are driven under cholinergic, so acetylcholine, you know, that is associated when you become awake. So in a certain way, you need some, there is an interaction of specific neurotransmitters, let's say, that will allow the neuronal network to engage in 40 hertz oscillators. Then there are, of course, theories. Andreas Engel was proposing that these 40 hertz oscillators are the big building blocks of brain function in a way, because they can be explained, they can be generated in vitro. So in theory, I don't know how it extends to all the cortex, but at least, in other mammals, it has been found. So it seems to be really a property of the tissue itself, right? The property of the reciprocal connectivity between neurons. That's what is thought so far, because they have blocked different channels and things. So it's the feedback inhibition from inhibitory neurons that inhibit excitatory neurons, right? So the excitatory neurons fire, they will excite the inhibitory neurons, and the inhibitory neurons inhibit the excitatory for some time, and then it takes time, and it generates this rhythm. This is what is understood. But then the rest remains a bit, so there is a lot of theories on the corticothalamic loop that is important for the alpha rhythm, for example. And one thing that is important to say here is that in our model, one of the nodes is the thalamus. And we tried already also removing the subcortical areas, and then those time delays become shorter. So we have less of this collective rhythm. So for some reason, these larger loops from the cortex to subcortical areas may be involved again through this process, maybe a process similar to this. I think overall it relates to a bit to resonance, I guess.
Okay, thanks a lot. I have a second question, if it's okay for you. Yeah, that's very interesting. So I was wondering, if we have 40 hertz oscillator, can we explain the presence of higher frequency oscillation? For example, in epilepsy, we'll have high frequency oscillation in the brain.
Yeah, so there is also some interesting fact that even here I didn't show it too much, but when we have a system and some units synchronize together at a slower frequency, the others become more have power at higher frequencies, so they are more perturbed. So I think there is a an interaction maybe, and it may also relate with this fact that, so here the thing is, we don't know if these collective oscillations are actually doing anything, right? They are the alpha rhythms and that have been associated with idle rhythms or kind of inhibitory rhythms. So in a way, maybe what is interesting is not to see which brain areas are synchronizing in the alpha band, but see which are the other areas that become free, because they will be firing much faster because they are more frustrated. So they are not being able to keep in their limit cycle, and so they also oscillate. So yeah, the full understanding of this dynamics isn't clear, but yeah, I don't know. And also maybe the faster frequencies may be related more to the alterations in neurotransmitters, so really in the physiology of the neurons. But in that sense, I'm not really familiar if there's any experiment showing in vitro higher frequencies. But indeed, in task, it is known that there are high gamma. But that was something interesting I found, that in this range where you find the collective rhythms, there's also seems to be more power at higher frequencies that may deserve further exploration.
Thank you. Okay, thank you very much. Any other question from the audience here? No, I have a question. You have one?
Yes.
Okay, go ahead.
So thanks for the very interesting presentation. You mentioned the fact that for complex and very large network, you might have synchronization, well, not global synchronization, but local, like some clusters. So it is known that in such complex network, symmetries also is also linked to local symmetries, local synchronizations, right? So I wonder if there is any study of actual data that shows such synchronizations that are provoked by some symmetries and in functional networks.
Usually on the work on functional connectivity with MEG, for example, they found that contralateral areas in the brain systematically like increase their power at the same frequency simultaneously. So for example, auditory areas, they show for visual, auditory, somatomotor, like for different areas. I don't know if this is, but again, I don't understand here if the symmetry is in the topology of the network itself, or for example, in the coupling, there is another thing that is here. It really comes from the symmetry of the coupling. So this area is coupled to this with equal strength as this is coupled to this, right? So we assume bidirected coupling. And indeed, there are many theories that say that no, the signals that we see are due to the fact that there is more directionality in the couplings. So indeed, you can create loops by loops, or let's say the systems, by breaking this symmetry in the coupling itself. I think all of those properties are really worth exploring. Unfortunately, I'm not really familiar with a paper itself that I can link to that, but I'm sure, because I've been hearing these discussions about the importance of certain features. So what are the key features of the network that allow for this, or of the dynamics? In terms of the symmetry itself of the brain, it's a bit ambiguous, because some people can live with just one hemisphere and all these things. But more in terms of the coupling itself, I think there's still a lot to understand. To be honest, I'm still lost. Though they say I contributed a lot to the field, I feel like I also contribute to bringing even more questions. As you'll see, I don't know, but probably.
I mean, the studies about symmetries and synchronizations I've came across so far are all very theoretical and mathematical. So I've yet to find something really concrete about this. So yeah, so thanks for your answer.
No, but exactly what you raised, this point about the research that exists, it was something I faced also through my PhD, and that when trying to use the Kuramoto model and applying it to this, then trying to understand what is going on, I had to dig into papers from physical review letters that, as you say, are very theoretical and sometimes, at least from my side, not approachable from my background. But so it requires more translation between fields, across fields, right? So really our collaboration across fields so that we can test. That is the thing. Even if I cannot prove, at least I can run the simulation. You can try to break the symmetries and that and run the simulations to see if then it links to the works that are done theoretically. Because for the theoretical mathematicians studying complex networks and that, it's also really nice for them to have an application for neuroscience, right? Yep. So it's a two-way gain, and I think it's very important to promote, yes, and then not to be afraid to merge these fields and understand better what is going on.
Yep. Yeah, good.
Okay,
Yeah,
thank you very much. I have few questions who comes online, so just I try to summarize few of them. Because we had a very nice talk given by Maxime Descoteaux in how we got the structural connectome, what are the difficulties. So the question is related to what he says, and the question is how the dynamic change depending on the error that we make on the structural connector. So because he said that, okay, what you see is not the reality, and there are a lot of error on the connectivity between population of neurons, how this change the dynamics. So
yeah, that is a very good question. And to be honest, yeah, for example, during first for the Kuramoto paper, I had the structural connectome from 22 subjects, and I decided I was running simulations with the average from all of them, right? But then I wanted to see how it changes if I just do with the individual connectomes, right? To see how this, so what we find is that this parameter space, the shape of this parameter space qualitatively is the same, but then the transitions to synchrony, the different borders, there are slight differences that could in a way explain why the best fit to each individual subject here was found by changing mainly the conduction delay and the coupling strength. But of course, just changing a bit the connectome, which I think aligns with also the differences, can give certain, can explain the variability that we find. Another thing is when we change the parcellation scheme. So here, for example, for this paper, we use the AAL parcellation scheme, but then in the supplementary information, we show with the Schaeffer parcellation, which is with 200 areas. So to show that qualitatively, okay, so the shape of this parameter space, the emergence of collective oscillations, this holds, right? There, it's a common property. Then in the first Kuramoto paper, then I perturb the network in different ways, right? We can, instead of having heterogeneous couplings, I put like all-to-all coupling. If you have all-to-all coupling, everything synchronized. The border to synchrony is super sharp. So there is no, 1 second. I was running out of battery. The transition to synchrony is very sharp. Also, we tried what happens if we have homogeneous time delays. And then we have less richness of different frequencies, right? The system is either synchronized in a frequency or in the coherent frequency, and we don't see for the same range of parameters, different frequencies. We tried, there's another paper where we remove nodes and remove links to the network to see what is happening, so how much it alters the dynamics. And for example, for schizophrenia, we show that if we remove link, so we were trying to replicate findings that were showing in graph theory, that in schizophrenia, the networks are more random in general. So there are different properties of that. And we could explain this by either removing nodes or links or just reducing the global coupling strength. So when we reduce the coupling strength, we will see more gamma and less alpha, right? Because there are less collective, and this is a feature of EEG in schizophrenia. So it's important, yes. And plus another thing is that the spatial fit between what we get from simulations and what is recorded, like the functional connectivity, there's always an error. We don't get a very good fit because as probably Maxime highlighted, the long range connections between interhemispheric, like same area in the two hemispheres, since they cross the corpus callosum, and there's a lot of it's harder to detect the fibers. So the connectome is never an exact match, although in the video I showed it and it seemed super accurate and high resolution. The reality is that there's a lot of variability, and the same, maybe Maxime also mentioned that. So if you run 2 times, so to the same subject, a diffusion image, and you compute the tractography, you can get different results. And this within subject variability is very similar to the between subject variability. So it's hard to know the ground truth. What we used here was a connectome, actually, from Andreas Horn. So we did the tractography using an algorithm that won the first prize in a contest of trying to infer the tractography, but also comparing with real tracing studies, right? So it was the one that best approximated the real connectivity network, but there's always this uncertainty. Yes, it's always it's never the ground truth.
Yeah, absolutely. So I think that maybe models in animals is can give some kind of input and some responses on that. In human is a little bit difficult. Yeah, I agree, but I believe that the structures, the synchrony that you observe is something that we can. It's a kind of features who are stable at the network level, even if we have some kind of error. But anyway, we have to demonstrate
qualitatively.
Exactly.
Qualitatively, because also our EEG is very different from person to person, even in the place of your alpha peak.
Exactly.
Yeah. So.
Exactly. So the second question who comes from, I think the same person is the he or she referred to a Bixby paper in Nature Communication, saying that the main fluctuation observed in the real data can be explained by the cortical structures and not the connectome itself. So is this something that you consider in your modeling? Because we see the underlying structures is the connectome. But if we refer to this paper, so the papers are, more importantly is the cortical connectivity.
Exactly, yeah. So that is, I mean, the cortical connectivity, you mean like nearest neighbor, or because we also consider the connectivity from one cortical area to another. But what I think is meaning is even the properties of each unit, let's say, or each brain area can change the local dynamics. So here we assume it's at 40 hertz, but maybe they, of course, and that is an oscillator, but it's a higher level of abstraction. But indeed, if we have more sensitivity, for example, to know the amount of excitatory and inhibitory neurons, because this is at least known from neuroimaging, that we can detect that from different contrasts in the MRI anatomical images. There's a contrast called the T1, T2 that is supposed to capture a bit the ratio of excitatory to excitation to inhibition in each brain area, and that will affect the local dynamics. And maybe at some scale, So I think it depends on the level of biophysical detail. And indeed, I never considered, because it's always, I'm more reductionist and trying to go to the most like, you know, you need to reduce at some point and to make assumptions just to focus on this principle in specific. But of course, adding complexity, I think there are different mechanisms operating. So this is not just the only one, and it will be a multitude of different properties. But the group from Michael Breakspear, at least there is the Fryer paper from 2010, they show that they have the cortex and then they have the thalamus, and it's the time delays between the cortex and the thalamus drive the 10 hertz oscillations. So from time to time, the cortex is decoupled from the thalamus, so it will be at 40 hertz, and then it couples with the thalamus, and you see a 10 hertz oscillation, then it decouples. So this would induce fluctuations in the power of alpha rhythms, but in a way it is very related to this principle. It's just a different way to look at it, but I think it's the same phenomena that is described. Yeah. So we can always add more pieces.
Absolutely. So maybe one of the things that we can consider is to add another network, which is the structural or cortical networks, adding this with the a connectome that we have. So we have, rather than having one network, we have two anatomical networks. So
but what do you mean about the cortical network is what I'm confused, because for example with the Schaeffer it was only cortical areas, the connectome, you can do a connectome only. So you mean like through space, like the connectivity?
Yeah,
more...
through the cortex. So we have like one of the things that we had a nice presentation yesterday about the role of the astrocyte and how the astrocyte can control the population of neuron to fire or not. So, and if we look at, this is a simple hypothesis who comes from Hugues Berry, who did his presentation. He is coming this afternoon. He's what we see from the astrocyte point of view, the astrocytes are kind of are related by the gap junction, and this is, we are related to the neighbors related to the neighbors. And if you map...
so neighbor to neighbor, propagation through space and not through these long wires.
Absolutely.
Yeah.
So
and there are some groups already doing this. So considering both the nearest neighbor connectivity or propagation through space, let's say.
Absolutely.
And my talk in this afternoon also puts all of this to question and is more aligned to that. And I showed I don't know if the paper they were mentioning from Breakspear in Nature, if it's the geometric constraints on human brain function. I think then it's not Nature Communication, it's just Nature. But I will show that this in the next session, and how I'm finding evidence in fMRI that it seems to be in that direction, but still for EEG, I think it's more through the axons. I don't know. It's very interesting, but it's indeed this approach of maybe there are different things interacting together, different types of networks, giving rise to different signals that we detect from the brain.
Maybe following the question is that if we introduce this kind of constraint coming from the exercise, maybe this can explain the delay that you mentioned on the dynamic system. So that's maybe something that you put the delay, and the question is, okay, where this delay come from? So one of the explanation is that all the area are not activated at the same time, which is completely true. But we have also, and you mentioned this about the neurotransmitters, and so depending on the concentration of the GABA and glutamate within some areas, so the firing rate change and the population of neurons change. So there are more synchrony observed when those concentrations are different, and they are different in different areas of the brain. So, and this may be can be the underlying physiological process who can explain the delay that you introduce in your model.
Yes, well, the delay here is the conduction time. It takes
Yeah.
time to propagate long distances, but and it's in the order of the realistic time delays that are believed to take. If you perturb here, it takes some time to reach the frontal. If you perturb the visual field to reach the frontal eye field, around 10 milliseconds. But so that was here. But indeed, there is so the surface models, let's say, so there's always a time associated to the distance in space. Either it's through the axons or through continuous space. And indeed, also the fact that here the coupling is symmetrical and we use this phase coupling, which is not an excitatory coupling, suggests that maybe there is a propagation of these local fields through space, but maybe having more realistic models or testing the two. So what the others did, they infer the model, the modes. I think it's a different property here because, yeah, addressing fMRI or addressing EEG, and also addressing the oscillations, because in a way, from what you're saying, even if it was the glutamate changing, like, why would it simultaneously drive different brain areas? So a change in those neurotransmitters, what would make them simultaneously increase in distant brain areas, right? And why would that drive a slow rhythm? Like, where would the slow rhythm come from? So there are some yeah, but all those things are very dynamic and they're always moving. So I think we really need to understand better these dynamics.
Yeah, I have some evidence that yes, the changes in the metabolism, and mostly in the neurotransmitter GABA and glutamate can shape some areas related to what we call network synchrony, and this network synchrony comes mostly from the increase of the glutamate in different areas. For instance, we have some experience with Julien Doyon at McGill University, looking at how the brain learns. And the first learning, we know when you do some sequence, motor learning sequence, you are involving a specific network. And this specific network, if you look at carefully on the area who are involved in this specific network, they change over time the concentration of the GABA and glutamate. And this concentration means that, okay, when we stimulate the brain, the most area who are activated or mostly activated, this is the network. And this network seems like heavily related to this underlying physiology. So
okay, so but the glutamate would be driven, like the increase in glutamate is driven by what? By the task. So the task would drive.
By the task, yes.
Yeah.
And
the task would drive
absolutely.
a release of glutamate in those brain areas, and then they would become active. Maybe then they would fire at 40 hertz.
Absolutely.
And then they would synchronize together and drive a slow rhythm. Yeah.
Exactly. So there is like an underlying process who seems like merging between what is going on at the level of the what the astrocytes are doing and what the neurons are doing at the same time. There is like
exactly. And now that you raised this point, this is a thing I'm also like, here we perturbed everything with just uncorrelated white noise, but of course things are not being perturbed just with uncorrelated white noise, right? Like I said here, this is what drives these fluctuations in LFPs, let's say, or the firing rates or whatever. And what is this noise is not noise, it's physiology. So I'm sure that having more adequate models, maybe then we'll have a better fit, because a change in those properties of this noise, let's say, can really change the dynamics indeed. So yeah, so there's a lot to explore in that direction.
I think it's a very interesting question. So anyway, I have one last question from the audience. Is you show the origin? What is the origin of the fluctuation seen on the envelope of the MEG data in the alpha and beta band. So you show this. So do you have any idea where it come from?
So it is so the fluctuations in real data, we see
In
those
real data.
fluctuations and we don't know where they come from. In the model, the fluctuations here were driven by spontaneous, so it's called metastable synchronization, which is when the system is not, when the coupling is not sufficiently strong to fully synchronize. What we observe is some subunits synchronize, then other subunits synchronize, and this is without noise. So it's a property of complex networks. They just stay there, synchronizing a bit, synchronize. So I show here, this is 250 seconds. You see there are periods of synchrony that can last. This depends on the coupling as well. If I put stronger coupling, the stability of the synchronized clusters is longer. But yeah, in the model, I don't know about in the real MEG data. I see them, but I don't know. This is just a possible explanation that is associated to transient synchronization.
It has something to do with the topology underlying the network?
Exactly. Yes. If we put all-to-all coupling, you don't have, so the transition to, so what is important is that you need, when you simulate, to have this border to synchrony where the synchrony degree is low overall. It's like 0.2 if I do the average synchrony degree of the system. I mean, again, this is just 40 hertz oscillators coupled together in a given network structure, right? And then we increase the coupling and we see that some of them synchronize and desynchronize. And when we have these fluctuations in the synchrony degree, so this is the standard deviation of the synchrony degree over time, right? Is when we find this the emergence of these oscillations, of these collective oscillations. So it's neither fully stable, so we need this transition border. And all the human connectomes that I tried have this sharp border, but I also have simulations where if I don't have complex network structure, so if you just put all-to-all coupling, or if you just this border to synchrony, it's like it goes, it's all dark blue, and then suddenly you have a border to synchrony and everything is red. Like you have either zero synchrony or one. It's not, you don't have a middle. So this is there is a paper, the Young and Strogatz 2009, I think, where they describe, for example, for the first time, these non-steady order parameters. It is what is called. So this the order parameter. So the order parameter is the level of synchrony. So it's neither fully synchronized nor fully synchronous. The system is really fluctuating between synchrony and asynchrony. This is something we put here. Like this is the level of synchrony over time, and it spontaneously fluctuates. But here we had a bit of noise. In the Kuramoto, we didn't have noise. So it's not noise that is driving this. It's just these competitions in the system that have been described. I mean, in the literature of the systems, I think I put here some papers of the theory, and in the metastable chimera states in community structured networks, they didn't have noise as well. So it's really, it just stays for it's a non-stationary dynamics. Mm-hmm. It's very interesting, but there's nothing driving it. It just appears from the connectome structure because all units are identical. They're all 40 hertz, mm-hmm. Right? There's no noise. So the only thing that is, so it's kind of frustration mechanism. So one synchronized, but then there's always others that do kind of a competition that destroy that synchronization, and another will synchronize. So it allows the system, and there are several papers, a number of papers again in very physical reviews, and that describing this non-stationarity in the border to synchrony. Okay. So below in the subcritical range. So there's a synchrony and for a coupling that is below that. But yeah, I mean, I don't know exactly, but that's the thing. This is a self-organizing principle. It's an intrinsic thing. There's nothing else driving those oscillations. It just, that's why we call emergent.
Okay. So thank you very much for all
You're
those
welcome.
explanation. We are going to have like a small break, and we are back at 11 for the next step of the presentation. Okay?
Okay.
So thank you very
Thank
much.
you. Thank you so much. See you later.